Hi class, welcome back. We're finishing chapter 6 now. We've talked about how you grow bacteria on a plate. We've talked about how we make the media, how we culture the organism, how it goes through a growth curve. But now we're going to talk about how we count it. There are some different methods. Some are direct. A direct method means you are directly counting the number of cells there. Some are indirect. That means you're measuring something else and correlating it to the number of bacteria there. So the first way we can count how many bacteria are there is using a filter. We'll filter out the bacteria from the solution. So if you are wanting to see if there's bacteria growing in your milk, you're a dairy farmer trying to sell your milk at the market, or you are an environmental scientist wanting to see if there's bacteria growing in a lake. You can take a sample of that milk or that lake water and pass it through a filter. When it passes through the filter, the water will go through, but the bacteria will be stuck on that filter. Then you take the filter paper, here this grid is the filter paper, and you put it on top of your culture media, and the bacteria, if there's any bacteria stuck to the filter, it'll start growing. Every little bacteria that grows will show up as a colony, and then you count the number of colonies there. And each colony will tell you how many bacteria you started within your solution. Next is doing a plate count. In a plate count, you're going to count numbers of bacteria on a plate. You can either do it in a pour plate method, where you mix your inoculum, whatever solution has a bacteria in it, mix it right into the nutrient agar and grow it all together. In this situation, you'll have bacteria growing in the media and on top. Or you can spread your bacteria, you can pour your bacteria, spread it around on top of the agar. And in this situation, you'll only have bacteria growing on top. Either way, you're going to then count how many dots are there, and you'll know how many dots were in your initial solution. Sometimes there is a lot of bacteria present, and you can't just pour it on top. You'd have way too much. So you're going to have to dilute it. So if your milliliter of milk had 10,000 bacteria, there is no way that you could count out 10,000 colonies on a plate. It would be one big old smear. So you could dilute it. If you diluted it to 1/10 of that, you'd have 1,000 bacteria. Well, that's still a lot to count. If you diluted it to 1/10 of that, you'd have 100 bacteria. That's a more normal place to count. So you take your inoculum, you dilute it 1 to 10, 1 to 100, 1 to 1,000, 1 to 10,000, 1 to 100,000, and then you take a milliliter of each of these samples and pour it out onto your plate. At these solutions, you can see way too many to count, so you cannot figure out how many bacteria are at these solutions. At this dilution, there's not enough. If you're only counting 4, what if that's 1 or 2 too many or 1 or 2 too few? If it's just a little bit off from average, you've wildly changed your number. But this solution, you want it somewhere between like 20 and 200 or 250. So that's a nice number to count. Here it says there's 32 colonies on the plate. So then if you know you've diluted it this many times, you multiply back by that and say there's 32 times 10,000, you have 320,000 bacteria. Serial dilutions, I want you to take your time and think through this, how that's working. I want you to make it make sense to yourself, because this is also a question that very frequently pops up on your entrance exams to get into nursing school. So I want you to be able to understand this concept. If you need to come talk to me, that works. We will be doing this in lab 8, I think in lab 8, and you'll get a little bit of hands-on experience. It makes it make a little bit more sense. Okay, another way to count your bacteria directly is by a microscope. So you just take a little sample of your solution, you measure a specific amount, pour it onto a slide, and use a specific type of slide that has this grid markers on it. And using the grid markers, it helps you keep track of what bacteria you've counted or not. You count how many bacteria are in that square and multiply it back to get how many bacteria were in the start. Then there are indirect methods of bacteria growth. And an indirect method, you're measuring something else, but then you can use that to compute back to how many bacteria you have. One indirect method is turbidity. So do you remember when we were practicing aseptic technique in lab? If you had a tube with no bacteria in it, it's nice and clear. But as bacteria start growing, it gets a little bit cloudier. So less light is going to pass through it. You can use a spectrophotometer. That's a big word that you don't have to remember. But it will take and shine a light source through it and measure how much light is going through it. You can compare that to just a tube with water to get how much light could pass through it if there's nothing there. Compare that to how much light is passing through your solution. And it'll give you a reading of how much light passed through, how much light was blocked by your solution, how much bacteria you have. You just multiply it back to get how much light is in there. You can measure the dry weight of your sample. So you dry it out and you weigh it without the water, and that weight can then be used to multiply back out to get your original concentration. You can do most probable number. This is something that makes more sense when you do it in lab, but you're going to have a bunch of test tubes, and you're going to just do an experiment and see how many bacteria react. And you'll see how many bacteria react, and then just count up how many tubes. So here all turned yellow, they all reacted. Here 3 of them turned yellow, 3 of them reacted, and here 1 turned yellow. And this, none of them reacted. So you just compare how many reacted. You just look at a table and it tells you if your number is 531, this is how many bacteria you have. Okay, direct methods usually require an incubation time for a colony. Why would that not always be feasible for analyzing foods? Why would you want to be able to say, right now, this sample has this many bacteria instead of, well, 2 days ago, this sample had this many bacteria? Okay, that finishes chapter 6. I will meet you back for chapter 7. See you later. Bye.